So what you should be seeing here now is the title screen. And prior knowledge, things that you should know from your studies of biology at Key Stage 3, that's years 7 to 9, is that you should recall that enzymes speed up chemical reactions, what they're for. Recall and identify some enzymes used in human digestion. And finally, you should be able to recall and describe how substances can move by diffusion. That's not going to be covered in this one, but by the end of this lesson, what you should be able to do is describe an enzyme as a biological catalyst and what this actually means. Define the term monomer and the term polymer with examples from human digestion. And finally, describe with an example the problems that can arise if enzymes are not present or don't function correctly in living organisms. So our first outcome, what is a catalyst? Many chemical reactions, just talking about chemistry now, can be sped up using a catalyst. The idea is that a catalyst is a substance that is not used up in the reaction, but it makes the reaction go faster. So in this case, we've got our two carts at the bottom. The catalyst is the one that's sat on the red reaction cart, and it makes it go faster than the blue. At the end of the reaction, though, the catalyst is left behind. It's not used up in the reaction, which means that you can recover it, and it can then be used for the next chemical reaction to help speed up the reaction again when needed. This is quite important because it means it's always present and always there if you need it. So a biological catalyst then, how is that different from a chemical one? A biological catalyst is a molecule that's found in living systems that can speed up the reactions necessary for life. You're a walking grab bag of chemical reactions, and if those chemical reactions slow down or don't work correctly or don't work at the correct speed, then it can affect your ability to process energy, repair yourself, fix damaged DNA, and all these things are controlled by chemical systems. In a living organism, the catalysts that speed up the synthesis, or the building up of, and the breakdown of chemical molecules or biological molecules are called enzymes. This then means that enzymes are biological catalysts, and like chemical catalysts, they increase the rate of reactions, but in living things. In short, enzymes are biological catalysts that can speed up the rate of a biochemical reaction. And that is our first learning outcome. An enzyme is a biological catalyst. Second outcome then, monomer and polymer, examples from human digestion. Before we go on, a little more about enzyme reactions. In the same way that a chemical reaction has a reactant and a product, enzyme reactions talk about substrates and product. In reactions, the substrate is what the enzyme starts to work on. And remember, it can either work on it by joining small substrate molecules together to make larger ones in a process we call synthesis, a bit like taking lots of individual Lego bricks and building a bigger wall, or breaking larger substrate molecules into smaller ones, digestion, kind of the reverse, taking a Lego wall and breaking it down into individual bricks. Whereas the product is the same as the product of a chemical reaction, and this is what is produced or made during the enzyme catalyzed reaction. So let's get into what a polymer is then. A polymer is a molecule that's made of lots of repeating units called monomers. It sounds a bit complicated when we say it like that, so the easiest way to do it is to visualize it as something different. Before we get there, the word monomer and polymer actually come from, well, there's an etiology to the word, and mono means one alone, whereas mer means of a group. Poly means many, whereas mer means of a group. So if we imagine this a bit like a jigsaw puzzle, with lots of pieces joined together to make the whole picture, the monomer is every individual jigsaw puzzle piece, whereas the polymer is all the pieces joined together to make one big thing. That means we can build polymers from monomers, or we can take a polymer and break it down into individual monomers. In digestion then, humans, the process of breaking down substances in our food into smaller, soluble or dissolvable molecules is called digestion. Different digestive enzymes are needed to break down the different nutrients in our food. But to be fair, the job of each one is to work on a large substrate polymer molecule So lots of repeating units joined together. And break it down into either smaller polymer or individual monomer product molecules. Let's have a look at what this means with some specific examples from the human digestive system then. Starch. Starch is a polymer. It's a storage molecule for energy. If you like, it can look something a bit like this. Lots of the same monomer unit joined together. I mean, this is a very shortened version of a starch polymer molecule, but it essentially is the same idea. It's a repeating pattern of the same monomer joined together by chemical bonds. In this case, the repeating monomer is glucose. Starch is a long chain of glucose molecules joined together with chemical bonds. Enzymes, such as amylase in our saliva and in our small intestine, they act a bit like scissors and break the bonds between the starch polymer molecules to release the individual glucose subunits. So there's our scissors going off, cutting that bond, and what we get released is smaller glucose monomer molecules one at a time. The polymer starch will not break down at the same speed as it does without amylase. So amylase is important to get that reaction to happen at an appreciable speed that we can release energy from our starch. Digestion of protein. Protein is also a polymer. Protein polymer molecule shown here. Now, proteins are made up of something called amino acids, and there are 21 different amino acids that can make up a protein. A protein can be any sequence of any of the amino acids put together, which is why I've shown every individual subunit as a different color. It's a long chain of amino acid molecules, again joined together with chemical bonds. Enzymes such as protease, in our stomach and small intestine break down the protein polymer. Same action as before, they snip away at those chemical bonds, and what they release is single amino acid monomer molecules. There are 21 different amino acid monomers that can be found in a protein polymer chain. Final example from digestion, lipids and fats. Lipids are a polymer, and now these are a little bit strange. It's a monomer of two particular molecules. They're a long chain of fatty acid and glycerol molecules joined together with chemical bonds. So the fatty acids are represented by the zigzag tail and the glycerol molecule at the head. Enzymes such as lipase for lipid, and ase is the name of pretty much any enzyme that breaks down a subunit, break down a lipid polymer, but now it's snipping it there. And what we get is smaller fatty acid and glycerol polymer molecules. Synthesis. The previous examples were examples of digestion, of enzymes being used to break down large polymers into smaller monomers or smaller polymer molecules. Synthesis is the opposite. It's the building up of smaller monomer or polymer molecules into larger polymer molecules. Again, using the example we mentioned earlier, it's taking Lego bricks and building them up into a Lego wall. In humans, the process of building smaller molecules into larger ones is called synthesis. Different synthesis enzymes are needed to build up the materials needed for our body, but again, each one works on a small substrate monomer or polymer molecule and builds it up into larger polymer product molecules. Once the smaller products of digestion are absorbed into the body, they can be used to make larger molecules to build new cells and tissues. You might be wondering what the link in this is, but the digestion of the foodstuffs that we eat provides raw materials for our body to then build new things that we need. We can't take everything we need from our food, but we can use the raw ingredients to build it, which is why we need enzymes that break down and enzymes that build up. Starch. Starch, again, is a plant storage polymer that plants use to store excess energy from photosynthesis. Enzymes such as starch synthase in plant cells form bonds now between the smaller glucose monomers. They're not breaking them. It's kind of the reverse reaction that we had with amylase before. So there's our glue going in, starch synthase acting like a glue, and it's bonding those single glucose monomer molecules into larger starch polymers. This larger starch polymer is then stored in the plant until it's needed for respiration. So why use the enzymes again? Well, the digestion or the synthesis of molecules in the body happens very, very slowly. I mean, really slowly, like you're talking on the orders of, in some cases, millions of years. Even then, for it to happen, you need the correct amount of energy to break those bonds between the smaller subunits in digestion. Or if you're trying to build things up, you need the first piece and the second piece to come together in the right orientation and with enough energy to bond. The way I liken this is that you could take a Lego brick and another Lego brick, but if you don't have the bottom of one lined up with the top of the other when you put them together, they won't stick, they'll fall apart. That's the similar sort of idea with this. Enzymes speed up the process of these reactions tremendously. They help to either provide the correct amount of activation energy for the reaction to happen, or even in some cases can orient the substrates in the right orientation for them to react faster rather than waiting for them to just line themselves up. They can turn very slow processes into one that happens in less than thousandths of a second. So it really does speed things up. As an example, there are about 3,000 different enzymes in the human body that catalyze reactions which otherwise wouldn't normally occur. OMP decarboxylase helps produce a substance that makes DNA in your body. Now, DNA is really important because it contains all your genes and genetic information. It's produced in 0.0018 seconds with this enzyme present. Without it, the reaction would take around 78 million years, meaning that life would not have started. So, the term monomer and polymer with examples from human digestion. Final bit then. Describe, with an example, the problems that can arise if enzymes are not present or don't function. Starfish. You might not be able to see this here, but in that picture there, from the center of the starfish, there is a protuberance, and it's not the starfish's leg or arm, if you like, but it is in fact its stomach. A starfish feeds by pushing its stomach outside of its body and onto the food or into the food that it's trying to eat. It then releases digestive enzymes to break down the food and absorb it. If the enzymes are not made or don't work correctly, the starfish would starve because the food that it's trying to digest would remain undigested and the nutrients it could not take in. Second example. You may have seen this yourself if you've had a baby brother or sister and you've been to the hospital. Just after a birth, within the first few hours of life, there is something called a heel prick. An infant heel prick test seems like a cruel idea, but actually it's a way of testing the baby's blood. They're just basically pinprick with a small needle, sterile needle, on the baby's heel, and they dab a spot of blood onto circles on a piece of paper, and that blood is then tested. One of the substances that they test for is the enzyme phenylalanine hydroxylase. This enzyme breaks down the amino acid phenylalanine, which is found in food. If it's not broken down correctly, then the phenylalanine can build up to excesses in your body, and without that enzyme, the child will grow to have nerve and brain damage because they can't break it down fast enough to prevent its effect in large amounts on the body. Final example, cystic fibrosis. Now, you may have come across this already, you may even be living with someone, or you may have the condition yourself. Cystic fibrosis is a disease that's caused by genetics. You have a copy of a recessive genotype, which in turn causes you to have a faulty carrier transport protein, and it means mucus builds up in all of your respiratory system, your ducts, everywhere else. It's also known to cause malnutrition amongst other more well-known symptoms, such as fluid building up in the lungs. And this is because the ducts in the digestive system that transport enzymes to the food as it's passing through are blocked by mucus. This then prevents the person from being able to digest the nutrients from their food as quickly as a non-cystic fibrosis sufferer. And because they can't digest the nutrients as quickly, in a very real sense, it means that they aren't absorbing those nutrients. They can't then go on to make new materials for growth and repair. So they suffer from malnutrition, even if they're eating the same diet as a non-cystic fibrosis sufferer who's perfectly well, shall we say, able to absorb the nutrients that they need in their diet. They will still suffer from malnutrition. In some cases, then, this needs to be treated with oral enzyme supplements to help them start breaking down the food as it's passing through the body chemically before their body starts to work on it as well. So that's our final learning outcome. Describe with an example the problems that can arise if enzymes are not present or don't function. Thank you very much for listening. I hope you've enjoyed this lesson, and I hope to see you again next time. Thank you.